Good morning to everyone. It's 10:02, so it's Thursday morning, means it's Enjoy Science time. I want again to thanks to all of you who are coming here. As you can see on these slides, large numbers. We are happy to have over 10,000 attendees to Enjoy Science during these 18 months of wonderful talks. Of course, I want to thanks again to Liz Kent and to Emily Lang for their wonderful work to organize it, make my life and the life of Alessandra Ferraioli, we host together Enjoy Science very easy. It's my pleasure today, of course, to introduce Dr. Sharon Dent. Everybody knows Dr. Dent. She is a professor in the Department of Epigenetics and Molecular Carcinogenesis, the chair of the department, and this is a way to welcome her back in Houston. They just transferred from Smithville, so welcome again in the mainland of MD Anderson, and thank you for everything what you have done and you are doing for all of us, because you know very well Dr. Dent work it's on an exciting topic of epigenetics, and her publications and her group publication are in the most exciting journals, and all the time are wonderfully done and wonderfully taught research. It's very nice and very helpful for all of us to read and to learn from Dr. Dent science. With this, please, we are all very happy to have you here, and we are all very curious for your talks. Thank you again, and good luck with everything.
Thank you very much, Dr. Kalin, and thank you for the invitation to participate in this series. As you said, we just moved from Smithville to Houston, and that on top of the COVID pandemic has really impacted, I think, every research program in the department. So it's nice to take a breath and remember why we enjoy science so much, and focus more on our curiosity, our innate curiosity, and the problems that we are looking to solve. So I titled my talk Lessons from the Saga Complex, because in putting the talk together, I just thought about some sort of high-level lessons that I take away from our work, and I hope that as I go through the talk, it will be obvious to all of you as well. And these are probably obvious lessons anyway. But the first is defining protein functions in normal cells provides critical insights to disease. So often mutations in a factor are discovered in a particular cancer or a different disease, but it's very difficult to understand how that mutation is impacting the etiology of the disease or the outcome of the patient if we don't know what that protein normally does in that particular cellular context. And then our approach has been to use genetics to try to understand these functions, because genetics provides an unbiased view of protein and gene functions in vivo, and it can lead you to study things that you never thought you'd be studying. And I'll show you at least one example of that in the talk today. And another important take home is that histone and chromatin modifying complexes are multifunctional. It's rare that a complex is only a histone acetyltransferase or only an ATP dependent translocase involved in chromatin remodeling. Almost always these complexes are like Swiss Army knives, and they do many things. And so we need to understand all of the functions in the complex and how those functions work together. And that leads me to the last point, which is that defining the functions of even a single complex can take many years and many teams. And I'm very grateful to all of my teams. And I'm going to say thank you to my the team that performed the work today here at the very beginning, so I don't forget to do it at the end. And I'll try to point out as I go along who did what in the different experiments. Of course, I also am very grateful to the NIH for funding our research, and to all of our core facilities for enabling our research. Okay, so how did I come to enjoy chromatin research so much? What got me into this area? And I'm pretty sure it had something to do with looking at pictures like these back sometime early in my career. And I just couldn't help but wonder how it is that our genome can undergo such extreme changes in state between interphase and mitosis every single time a cell divides. And that really triggered my interest in understanding how these structures are regulated and how these different states impact DNA templated processes. So now we know, of course, that a lot of those changes in state are related to changes in post-translational modification of the histone proteins, which form the protein core of the nucleosome. And the nucleosome, of course, is the repeating subunit in chromatin. There are a multitude of post-translational modifications that occur on the histones, both in these tail domains that protrude out from the nucleosome and also within the core of the nucleosome itself. And each of these modifications has a strong regulatory potential in itself. And when you start to think about different combinations of modifications, it truly is a wealth of regulatory potential. And again, we want to understand how these different modifications function, both on a molecular level and also on a biological level in terms of processes and pathways. And just to give you an example, histone acetylation was actually one of the first modification, probably the first modification that was recognized as a hallmark for gene activity. It was recognized back in the 80s, maybe even the 70s, that genes that were highly transcribed were associated with highly acetylated histones, and genes that were less active or inactive were associated with less acetylated histones. And we now know that acetylation not only impacts the histones within a single nucleosome, it can impact how the chromatin fiber is folded into loops, how loops are organized into topologically associated domains, how those domains are divided into compartments, and how those compartments contribute to different chromatin territories. And even recent papers have shown acetylation and other modifications can impact liquid-liquid phase separation in vitro and presumably in vivo as well. So again, this is just one modification and a nice illustration, I think, of how it can impact a whole spectrum of chromatin organization. So our goals are to understand, again, the mechanisms of how these modifications are regulated and how they function in normal cells, with the goal of understanding then how alterations in those functions contribute to cancer. We're going to focus today on acetylation and ubiquitination because our favorite complex, the Saga complex, contains enzymatic modules that govern both acetylation and histone ubiquitination. So why are we so interested in Saga? Partly it's because of this protein, GCN5. GCN5 was the first protein identified to be a transcription-related histone acetyltransferase back in 1996 by Dave Alice's lab. We were very excited to be part of that collaborative team to make that identification. And that really broke open the field. Understanding that GCN5 was a histone acetyltransferase essentially provided a Rosetta Stone for identifying other histone acetyltransferases, and every single one of them that was identified had already been identified as something important in transcriptional regulation. And so that really cemented the idea that histone acetylation is not just a byproduct of transcription, but it might actually promote transcription regulation. So the other reason we're very interested in Saga is that it's highly conserved from yeast to people. I like this figure that I borrowed from a review by Jerry Workman. It shows the Drosophila and the human names for the proteins in the complex. Don't worry about it. You don't have to memorize these. I just like for you to take away that they're highly conserved, and the complex itself is very modular. And each of the modules is also highly conserved. So there's a module that performs histone acetylation. It also acetylates other proteins, of course. There's a module that is responsible for histone deubiquitination, and we've actually identified non-histone substrates for this module as well. There's an important module here that holds the whole complex together. And it also interacts with DNA binding factors, which bring the Saga complex to particular regions in the genome, because Saga doesn't bind to chromatin stably on its own. It has to be brought to a particular gene through interactions with sequence-specific DNA binding factors, and we'll come back to that in a few minutes. And then lastly, there's a module that interacts directly with the transcription machinery, and yeast helps to deposit the TATA binding protein, and it's suspected that this activity also is housed in the mammalian complex, but it hasn't been formally demonstrated yet. So already, here's one of the lessons I told you. This is a multifunctional complex. It's a little Swiss Army knife that does many different things, and each one of these 4 functions is very important. So I need to point out before I go on that GCN5 is also part of other acetyltransferase complexes, including the ATAC complex. The histone acetyltransferase module in Saga and ATAC are very similar, but the rest of the complex is completely different. And as I go through the talk, I'll try to explain why we think the functions we're examining are more Saga related than ATAC related. So what we know from studies in yeast and also in vitro and other cellular systems is that Saga, as I said, is recruited to particular genes through interactions with DNA binding factors. And once there, the histone acetyltransferase module decorates nearby nucleosomes, and this aids in the assembly of the pre-initiation complex for RNA Pol II. The deubiquitinase activity is important for the switch from transcription initiation into elongation. And to be honest, it's not exactly clear how that's happening. It involves histone H2B deubiquitination, but exactly how it triggers or helps to trigger elongation is not completely clear. So I had to put this in because early in 2022, groups published back-to-back papers in Nature showing the structure of Saga from just very distantly related yeast, and they're quite beautiful structures. We are not structural biologists, so this is not our work. What we can contribute to the field, again, goes back to the genetics. We've made a number of mouse and cellular models to probe the functions of both the GCN5 histone acetyltransferase and the USP22 deubiquitinase, and that's what I'm going to tell you about today. And although our focus started out in understanding what's happening during mouse development, and we've gotten some really interesting phenotypes in the course of our studies, as I'll show you, this really has pushed us into understanding how Saga contributes to oncogenesis. So much of our work has been focused on this single question: what transcription programs really require GCN5 in mammalian cells? And I'm not going to talk about our mouse mutants for GCN5 because they were published a long time ago now. I'm going to tell you briefly about a paper that we published that identified unique targets for GCN5 in mouse ES cells. And we did that, of course, by doing chromatin IP for GCN5 as well as RNA-seq. And this just shows you a snapshot of our chromatin IP. We used a biotin tagged form of GCN5, and these are basically all the controls. And you can see these beautiful chip sequence, chip signal for GCN5. When we looked at where those binding sites were, they clustered around the gene promoters as we expected. And then we did a motif analysis on these binding sites to try to understand what transcription factor might be pulling GCN5 to particular genes in the undifferentiated mouse ES cells. And we found these 2 very strong motifs, which turned out to be related to E2F1 and MYC. So it's not terribly surprising. There were other studies by ourselves and others that linked GCN5 functions to MYC functions in other contexts, but this was the first time it had been demonstrated so clearly in stem cells. So the RNA-seq confirmed that GCN5 is an activator. Most genes go down when GCN5 is removed. And then, of course, when we do the overlap between the ChIP-seq and the RNA-seq and what's known about MYC and E2F1 targets in ES cells, there was a strong degree of overlap. And the overlap genes are enriched in cell cycle and self-renewal. So this paper was published quite a few years ago now. So that's all the data I'm going to show you from that part of it. And the take home is that GCN5 is an important co-activator for MYC target genes in undifferentiated ES cells. And in the course of our studies, we also found that MYC directly regulates expression of GCN5 as well as several other Saga components. And that's why we think it's the Saga complex that's likely most relevant in this context. So MYC is a busy factor. It's not only involved in cell cycle regulation and stem cell self-renewal, it also functions as one of these so-called Yamanaka factors, meaning that when you add it to differentiated cells together with OCT4, KLF4, and SOX2, you can trigger those differentiated cells to lose their differentiated state and become more like pluripotent stem cells. So we've also asked whether GCN5 contributes to those functions for MYC. And this is an siRNA screen that was sort of unbiased. Callie Hirsch, the person who'd performed these experiments in collaboration with Jeff Rana's lab in Canada, had handpicked maybe, I can't remember now, it's either 300 or 500 different epigenetic factors and designed siRNAs to target those factors. So again, this was back in the around 2015. So that's why we were using siRNA. And she was using a system where you can induce the Yamanaka factors after treating cells, and in this case, these are mouse embryonic fibroblasts, with the siRNA, and then ask whether the cells reprogram. She monitored that by re-expression of alkaline phosphatase as a ratio to DAPI. And so the real experimental results are graphed down here. And just to give you an orientation, of course, if you knock down OCT4 or MYC, you don't get reprogramming. So or below the line sort of shows you where the loss of reprogramming cutoff is, and up here is the mock control. And this is just graphing out the impacts of the siRNAs for the different histone acetyltransferase, and what acetyltransferases in the set. And very clearly, you can see that GCN5, which is also known as CAT2A, popped out, but also 3 other components of the Saga complex were here below the line, showing that they had as much or more impact when they were knocked down as OCT4 and MYC. And just for comparison, P300, another very famous hat, is here. PCAF, which is a sister protein to GCN5, is up here with the MOX. So it really indicates that GCN5 and Saga are important for dedifferentiation triggered by MYC as part of the Yamanaka factors. And again, I'm just giving you the take home here, but what we found is that when you put the Yamanaka factors onto MEFs, one of the first things that MYC does is upregulate GCN5, and then GCN5 interacts with MYC, as we think as part of Saga, to turn on downstream factors. And in this case, the genes are not cell cycle genes. The genes are encode splicing factors, which are important for alternative splicing events, which promote dedifferentiation. I also want to just take a breath and say that MYC interacts with multiple histone acetyltransferase complexes. In addition to GCN5 and Saga, it interacts with P300-CBP. It interacts with the TIP60 complex. And in my mind, one of the big questions still out there in the field is, what's the division of labor here? I don't think, at least I would find it unusual, that MYC would interact with all of these complexes at a particular promoter. I suspect that there's some selectivity that's happening, and the big question is how and when and why. So that's a curiosity for the future that we can enjoy together. So given the fact that GCN5 is important for these two functions of MYC, it drove the question of whether GCN5 is also important for MYC functions as an oncogene. And we've examined this question both in lung cancer, studies of Lisa Mustachio, and also lymphoma. And I'm going to just talk to you about the lymphoma studies today. Why lymphoma? Well, one reason is if you look in Oncomine at different cancers where MYC is amplified, and compare that to expression of GCN5, again, which is also known as CAT2A, you see that in some cancers, there's this really high correspondence between MYC overexpression and increased expression of GCN5. And this particular cluster of overlap group corresponded to 127 cases of Burkitt's lymphoma. Burkitt's lymphoma, you may know, is associated with a translocation event that places the MYC gene under the control of the enhancer for immunoglobulin heavy chains. And this drives very high MYC expression in B cells, leading to B cell lymphoma. And also, these lymphomas are addicted to that high level of MYC expression. So another reason we chose lymphoma as sort of our test model for investigating how GCN5 might contribute to MYC oncogenesis is that there's a mouse model, the emu-MYC mouse, which mimics Burkitt's lymphoma in many respects. And this mouse contains a transgene which mimics that translocation that's found in the patients. MYC is being driven by an IgH enhancer. So what Amy did is to take the emu-MYC mice, cross them with a B cell specific Cree transgene and also our GCN5 flox allele. And so then she was able to create emu-MYC mice that were either wild type, heterozygous, or homozygous for loss of GCN5. And she simply asked, not simply, it was a lot of work. She asked questions about the survival of these mice and also development of lymphoma in these mice. And I'm just going to show you one data slide from this paper, which was published in the last year or so. And the black line in this Kaplan-Meier curve are the emu-MYC mice. They begin to develop lymphoma and die by about 18 weeks of age, and they're all gone before 40 weeks of age. The blue line are the heterozygous mice, and loss of one copy of GCN5 doesn't really impact survival or lymphoma development very much, but loss of both copies of GCN5 clearly extends survival. And in fact, when you do the numbers, you can see that the total loss of GCN5 almost triples the survival time for these mice. I'm not going to show you the data, but evaluation of the B cells, the lymphomas in the male mice relative to emu-MYC mice demonstrated that loss of GCN5 allows differentiation of the B cells to a further state than was normally found in the emu-MYC mice. So the lymphomas were somewhat less aggressive, if that's the right word to use for the mouse model. So that again provides some idea of how GCN5 is impacting the survival. So to get the molecular mechanism behind that, Amy did RNA-seq at a time point before lymphoma onset. And this is just showing you the heat map comparing 3 different wild type mice versus emu-MYC mice versus emu-MYC mice where GCN5 has been deleted. And I'll draw your attention to this cluster 5. These are genes that are highly upregulated in the emu-MYC mice relative to the wild type mice. And you can see that when we delete GCN5, expression of those genes goes back down to a state that's more like that in the wild type cells, although it's actually, in many cases, a little bit lower than what you would see in wild type. And we think that this is a combination of effects of changes in histone acetylation, because GCN5 and Saga are cofactors for MYC in driving target gene expression, but also GCN5 loss has a strong effect on MYC protein levels. And this is post-transcriptional. MYC RNA levels are not affected, but the protein levels are strongly decreased when we delete GCN5. So our work, together with other studies from other labs and our work in many other contexts have led us to this feed-forward model. MYC itself is known to be a substrate for GCN5 and PCAF, as well as other hats like TIP60. And when it's acetylated, the acetylation of specific lysines blocks ubiquitination and thereby stabilizes MYC protein. It protects it from turnover. So stabilization of MYC by acetylation by GCN5 helps MYC work. And as I told you, GCN5 and Saga components are themselves gene targets of MYC. So MYC increase leads to increased GCN5 in Saga, meaning there's increased amounts of the coactivator for MYC, which increases the amount of target gene expression, including GCN5 in Saga, and so it's a cycle. So our findings are actually quite consistent with other studies that are coming out from other labs, and I find this quite rewarding. This is a nice study by Kim Stegmeier's group at Harvard, and they did a dropout screen for factors important for neuroblastoma formation, which is driven by MYC-in. And as you can see here in the top hits, she does get other hats like P300, but she saw 1, 2, 3, 4 different components of the Saga complex come out in her screen. And so we're working now with the Stegmeier group. Dr. Candy Chen in my lab is particularly probing the role of ATA2B in neuroblastoma formation driven by MYC-in. Another group shown here did a dropout screen for vulnerabilities in acute myeloid leukemias, and their top hit was GCN5. And I find it interesting here too that CAT2A or GCN5 inhibition induced myeloid differentiation and apoptosis. So to me, that's reminiscent of the increased differentiation we saw when we knocked out GCN5 in the lymphoma model. So more to come on that. Particularly, we're going to focus more on the ATA2B protein because that protein is specific for Saga. It distinguishes Saga from ATAC, and so we'll be able to more finely probe the functions of Saga in different oncogenic settings going forward. So in the last part of the talk, I want to turn to the other module, the other enzymatic module in Saga, the deubiquitinase module. It's, as shown here, it's comprised of 4 proteins. The catalytic subunit in yeast is called UBP8. In mammals, it's called USP22. If you just express USP22 by itself, it has almost no enzymatic activity. It requires these other cofactors to fold into the proper conformation to allow it to become a deubiquitinase. And we and others have identified important substrates for the deubiquitinase. The most well characterized is histone H2B. There's interest in USP22 beyond just transcriptional regulation because it was recognized many years ago now that overexpression of USP22 is linked to poor outcomes in a number of different cancers. It was first described as part of a death from cancer signature, predicting therapy failure. It's been associated with poor survival, poor prognosis in many different kinds of cancer. I'm just showing you a very few papers here just to show you the range of impact here. And more recently, it's even been associated with suppressing anti-tumor immunity. But again, how it's contributing to these cancers is completely not understood because the functions of USP22 in mammalian systems are just not well defined. So Leah Catello in the lab has taken on this question, and she has made a number of different mutant alleles of USP22 in mice. She's made a knockout allele, she's made a gene trap allele, she's made a catalytic mutation. And in all cases, it ends up causing loss of USP22, causes the embryos to die. And they die in an interesting way, unfortunately for them, but interesting for us. They are hypoxic, they're hemorrhagic, and they have many hallmark features of a defective placenta. So this is an example of a genetic approach that's taking you into an area that you may not have chosen to go into. If you'd asked me prior to these phenotypes whether I was interested in studying placenta, I probably would have said not really, no. But you know, once you get a phenotype, you become open-minded to studying all kinds of things. So Leah did a very wonderful job of further characterizing and defining the phenotype of these mice. And she found that the labyrinth layer in the placenta from the USP22 null mice was quite abnormal, was reduced. And the labyrinth is important because that's the part of the placenta where the fetal blood vessels interact with the maternal sinuses for nutrient and gas exchange. And if you take a step back and think about it, others have made the case that understanding placental biology is a lot like understanding tumor biology, because in both cases, you've got a situation where there's tissues that invade and manipulate neighboring tissues to set up a certain microenvironment. They have to establish a new blood supply, and they have to escape the host immune system. So Leah, again, I'm just showing you a smattering of her data. It's a really nice paper. I invite you to look at it. But looking at sections of placenta from wild type or null animals, you see these beautiful vesicular vascular structures stained by PCAM1 in the wild type case, and clearly abnormal structures in the absence of USP22. So how is this happening? To get some idea, Leah did a very careful RNA-seq analysis. She dissected out the labyrinth, and she did this at a very early time point before the phenotype became obvious. And the RNA-seq analysis was satisfying in that the gene ontology matched the phenotype we were seeing, even though we did this before the phenotype became obvious, but the genes that were enriched were important for vascular lesions, angiogenesis, vasculogenesis. And when she did upstream regulators analysis, she found many downregulation of many signaling pathways, including TGF-beta and multiple receptor tyrosine kinase pathways. And she confirmed that on the RNA level in the placenta, but she also took an orthologous approach in which she took mouse ES cells in vitro and differentiated them into endothelial cells, and then reevaluated their ability to form vessels, which I'm not showing you. Clearly, loss of USP22 impacted their ability to form vessels in vitro. But then when she looked at the RTK signaling in those cells, again, she found decreased phosphorylation of ERK, phospho-AKT, phosphocofilin. She found almost complete absence of some of the common intermediates for the RTK pathways, GAB1, GAB2, and she saw decreased intermediates, expression of the intermediates for TGF-beta signaling. She also saw decreased expression of some of the receptors themselves. So what this work told us is that loss of USP22 impacts signaling at multiple levels. Some of this is transcriptional because, after all, we started looking at this because of the RNA-seq. But we think it very likely that this deubiquitinase is impacting stability and turnover of some of these factors as well. And that's an ongoing area of research in the lab. So begs the question, are these same pathways affected in cancers where USP22 is overexpressed? Dr. Eva Chow, a former student in the department, helped us with this question. And this is just, again, one example. You can find many examples of this. So in liver cancers where USP22 is overexpressed, you do see upregulation of GRB2, SMAD2, GAB2. Many of the things that were down in the USP22 null mice are up in these USP22 overexpressing cancers, leading us to this sort of yin-yang working model, where loss of function of USP22 leads to downregulation of these pathways, impacting multiple processes. We predict that upregulation of USP22 or gain of function of USP22 would potentiate these factors, thereby helping to drive tumorigenesis. So nice model. Can you prove that? Well, we decided to test that by creating an overexpression allele of USP22 in the mice. And this was beautiful work done by Dr. Xinghang Kwang in the lab. And it was recently published. So again, I'm just going to give you kind of the take homes. We were very careful to analyze this allele functionally. I'm not going to go into details of how Xinghang set this up, but we created a situation where we can dictate when and where USP22 is overexpressed. And in our first experiments, we just decided to overexpress it in all tissues in the mouse from early embryogenesis on. And we confirmed, in fact, that overexpression occurs. We did biochemistry to show that it is incorporated into Saga. And we crossed it with our USP22 null mice to show that the tagged overexpressed allele is fully functional and is able to rescue the lethality of those null mice. So then we asked two questions. Is that overexpression sufficient to induce RTK signaling? And is it sufficient to induce tumors? So one of the interesting phenotypes we observed in these mice is an branching in mammary glands, as shown here. So these are 4-month-old mice. These are sections of mammary gland. And you can see in the mice that are homozygous for the expression allele, there's increased branching in the mammary gland. So Xinghang isolated mammary epithelial cells and looked at the signaling pathways that we noticed being abnormal in our other situation, but those same signaling pathways are important for mammary gland branching. And as you can see, when USP22 is overexpressed, we do see increased phospho-AKT, increased phospho-ERK, increased GSK beta, increased GRB2 levels. It's not affecting everything. We did not see any change in E-cadherin levels. So this says that yes, overexpression of USP22 can help to induce increased signaling through these pathways. However, that did not result in an increased tumorigenesis on its own. So Xinghang dutifully watched these mice over the course of their life. They basically died of old age. They had some spontaneous tumors, but nothing more than we saw in our wild type mice in the cohorts. Okay, so that's interesting. If it's not sufficient to induce tumorigenesis, and honestly, we weren't too surprised that it wasn't sufficient on its own, is it important for induction of tumors by these oncogenic pathways? So Xinghang did another round of experiments, and we've decided to focus on ERBB2 because of the mammary gland phenotype we saw in the overexpressing mice, and also because, again, there's a nice model for ERBB2 driven tumorigenesis in the mice called MMTV-NIC. Basically, these mice carry a transgene which drives HER2/neu expression from MMTV, and there's a Cree transgene just downstream. So it's physically linked, which means that whenever HER2/neu is expressed, that's where Cree is going to be expressed. And when you put that together with your flox allele, that it guarantees that you get deletion of your gene and overexpression of ERBB2/neu in exactly the same cells at the same time in the mammary glands. So Xinghang spent a lot of time creating these mice, evaluating them, and I'm going to show you one slide, which I think is a beautiful result, but it represents a heck of a lot of work. So mice carrying that MMTV-NIC allele begin to develop tumors about 120 days, and they quickly succumb to their cancers and are all dead by, about 160 days. Loss of one allele of USP22 does not impact that situation, but loss of both alleles of USP22 has a strong effect on the survival of these mice. We even had some mice that lived almost a year. And that increased survival is associated with a decreased number of tumors, also a decreased size of tumors, and decreased metastasis of the tumors. So now we are investigating the molecular basis for that decrease, and I look forward to telling you about that as we go forward. So I'm going to leave you with this sort of summary. And that is, again, by using our genetic approaches to understand the functions of USP22 and GCN5 in normal cells, that led us into asking questions about cancer. And we've identified processes and pathways that impinge on many of the classic hallmarks of cancers as defined by Hanahan and Weiberg. And I think this is certainly not a finished story, because we and others have noted connections between Saga, for example, and innate immunity, and also T cell biology. So I think there are going to be even more ways that this one complex, through its multiple functions, can impact these different hallmarks of cancer. And both of these enzymatic modules are potentially druggable. So I showed you a picture of the lab at the beginning of the talk. That was the pre-COVID, pre-move group. This is the group that has joined me here in Houston, and I'm very, very grateful to my lab manager, Andy Salinger, Leah Catello, Candy Chen, and Xinghong Kwang for coming to Houston, for being the core of my lab, and to taking our research forward to the next level. And I will stop there and be glad to ask questions, to answer questions. Thank you.
Thank you very much, Dr. Dent. It was a wonderful lesson of epigenetics in cancer and very interesting research. I have to say to everyone that usually the talks are recorded. I'm not sure if your talk will be recorded, but I invite everybody to go on Enjoy Science website, where most of the talks are recorded. And now let's start with questions. So Dr. Kapil Bala says, Sharon, fantastic talk. He has a comment: a dropout screen conducted in 2 AML cell lines show P300 and MDS, but not JCN5. So it may be AML cell line dependent effect. So if you can answer to this comment, and then I will go further with another question.
I think that's really interesting observation, Cap, and I'd love to talk more with you about that. Like I said, I don't think GCN5 is the only important hat. It's certainly not. It's just the one that our lab is focused on. So I, as I said earlier, I think the division in labor between these different acetyltransferases is an understudied area, and it doesn't surprise me at all that there will be cell type and even lineage specific functions for these different factors. And then I see you also have another question about may also be a cofactor and dependency in the double hit lymphoma. I'm not quite sure what you mean. Is GCN5 a cofactor for MYC amplification and double minutes? So we haven't looked at double minutes in particular, but I have not found a context yet where GCN5 and MYC are not co-amplified. So when MYC is amplified, GCN5 levels go up, and then the downstream targets also go up. So I'm not sure I've hit all of your question. Maybe we can talk more about the first part.
Now, thank you very much. Another question from my side. So, did you ever have the chance to check what are the long non-coding RNAs who are binding to this type of complexes, if they keep them on the DNA and all this function of non-coding?
I should have anticipated that question from you. I have to say we have not had a good chance to look at the non-coding RNAs. So maybe that's an area of collaboration for the future.
Thank you. Now another question, which is in chat, so I have to go between. So does tumorigenesis function of USP21 require other DAB cofactors? In human patients with high USP22 expression, do they also have high levels of other DAB cofactors?
This is a really important question, and we've actually tried to probe this ourselves, and it's not at all clear yet. So I can't give you an answer, but it's a very key question. Related to the question is whether the USP22 activity is required for these functions, and that we can address by putting in point mutations that destroy the activity and seeing if we get the same effect. We don't have mice expressing that point mutation just yet. In a live form, it's complicated because the mutation kills the mice as embryos, so we have to set up a special genetic way to induce that mutation while deleting the wild type gene. I don't want to get into too many details, but I think it's a very important question going forward. We have found that the cofactors that are required for activation of USP22 are also required for activation of two highly related deubiquitinases called USP27 and USP51. And so another very interesting question is, what is the balance between those three highly related USPs in oncogenesis, and how do they compete for these essential cofactors for their activity? So again, lots of interesting and important things to carry forward.
Thank you. Another interesting question from Dr. Anil Sood. So great presentation, Sharon. For the USP22 null mice, was the placental defect purely in endothelial cells, or was there defective pseudovasculogenesis, such as trophoblast line vasculature?
So Leah can correct me if I get this wrong, but we did not see much change in the trophoblast. We did see changes in both the parasites and the endocytes in the endothelial cells, and so that's where we focused on. So we do think it's more in the endothelial lineage, but we can't rule it out. And Leah is actually has gone on to use different Cree lines to try to probe more in more detail which particular lineages require USP22. So that's a good question. And while I'm answering your question, I'll say congratulations. I don't know if everybody knows, but Dr. Sood was elected to the National Academy of Medicine this week. And I think that's just a huge honor and well-deserved honor. So congratulations, Neil.
Yes, congratulations also from us. So thank you for letting us know, Sharon. So I think with this, thank you all. If you have other questions, please ask Dr. Sharon Dent. Also, if you are looking for t-shirts with Enjoy Science, please stay in touch with Liz Kent and Emily Lang. And with this, have fun, make discoveries, and see you all in 2 weeks from now. Thank you again, Dr. Dent. Thank you all.
Bye-bye.
Have a nice day. Thank you.
Bye-bye.